Okay, good morning, everybody. We're starting day 2 of the Marine Mammal Commission annual meeting in La Jolla, California, focused on emerging technologies to improve the efficiency and support innovation for marine mammal stock assessments. Welcome to day 2, where we'll have 2 sessions, one on reducing impact through innovation, innovative and less invasive sampling methods for health and population assessment. Then this afternoon, we have a session entitled Connecting the Dots, Innovative and Integrated Quantitative Methods for Assessing Populations. So welcome everybody here in the room and everybody online. For the folks in the room, just a few housekeeping details. Just let's keep this beautiful room clean, keep your lids on your coffee. Trash cans are located in the back of the room. The restrooms are out that door to the left, and that's also where the water bottle filling stations are. And the emergency exits are back there as well. In terms of the meeting, if you're online and you have questions, please use the Q&A function on the Zoom. And here in the audience, we have microphones to use. You just press the button. I think just a reminder that this is a public meeting. Under the applicable law, the Commission is required to maintain and provide a public record of the meeting, and as such, we will plan to post the meeting on our website sometime after the meeting. So our first session, Reducing Impact Through Innovation, is chaired by Dr. Randy Wells, a member of our Committee of Scientific Advisors. And I'll welcome you up to the podium, my friend.
Thank you, Peter. And I learned a lesson importantly yesterday. I learned this is actually a lectern, not a podium. Did I get that right? Okay, excellent. All right, thank you all very much for being here this morning, in person and online. The goal of our session this morning is to highlight some emerging technologies and sampling methods and discuss current challenges and future applications that could help to inform or improve stock assessments. Stock assessments, as we've learned and as many of us have known from experience, are extremely complex. We're going to come back down to earth a bit today from the fascinating discussions that we had yesterday about ways to get at one aspect, or maybe 2 aspects, of the stock assessments, abundance. There's much more to a stock assessment than just abundance. Being able to evaluate the threats the animals are facing, being able to understand the serious injury aspect of the animals, there's a lot to understanding even what a stock is and determining stock boundaries and making stock designations. Today we're going to be looking at, we're going to have 6 presentations this morning. They're going to show us ways that we can get at some of this information that used to be gotten primarily from hands-on work, now slightly removed, making it a little bit more feasible and easier to get some of the information. So I'm looking forward to all the sessions except this first talk, which is going to be mine. So we're going to be hearing from Chris Zadra on UAS-based deployments of suction cup attached biologging tags. Cynthia Smith will talk about a development of a minimally invasive blood collection device for cetaceans. Excuse me. Michelle Shiro will talk about harnessing emerging technologies for a non-invasive approach to marine mammal energetics. And the writing is too small. Ryan Kelly is going to talk about advances in eDNA monitoring of marine mammals, current capabilities and future directions. And then Ashley Barrett-Clough will talk to us about epigenetics, rounding out a very interesting session, I'm sure. So for the first presentation today, I'm going to talk about some work that we've been doing to try to change the nature of how tags are attached to animals. In order to be able to evaluate stocks of marine mammals, you need to know what they are. Biologically meaningful units are important when you can get them. When you can't get them, you rely on things like geographical cues or bathymetry or that sort of thing. But the more you can get the stock designations closer to the actual biology of the animals, the more meaningful the stock designation will be and the more effective will be the conservation measures that are used to try to deal with it. Telemetry is one method of trying to identify stock structure, for being able to identify the movements of animals through particular pieces of water. And it's been something that has seen a lot of development over the last 50-plus years. Today, on behalf of my co-authors, Michael Moore, Tom Lanigan, Jason Caput, Aaron Barleycorn, Jason Allen, and Robin Baird, I'll be talking about a method we've been working on for a number of years to try to be able to deploy tags in situations where it hasn't really been as possible before. So a lot of the telemetry applications to dolphins have been limited by attachment considerations. The available techniques for tag attachment have both benefits and drawbacks. One of the more standard ways of putting tags on things like dolphins is through hands-on, through catch and release tagging with fin mount tags. The tags can be secured with minimally invasive attachment. They can the risks that exist for the dolphins and the people from the capture and the handling process are a consideration. They can be logistically complex and expensive. They're typically limited to shallow water species or to species for which hoop nets can be used. But you can achieve attachment durations of months. The alternative to this for remotely deploying tags include the barbed projectile tags like limpet tags. These are simpler and less expensive than catch and release for deployment. You can access many species in habitats where shallow water catch and release is impossible, but there is potential for injury to the animal if the shot doesn't go as planned, and typically the attachment durations are less than desired. So our thoughts that went into coming up with a different kind of attachment technique, including wanting to be able to tag remotely without capture. This is for research and for situations where capture would be either challenging or logistically unfeasible. We wanted to be able to tag compromised individuals that would allow us then to monitor them further while making rescue preparations. We wanted to potentially tag members of large groups that may be in the process of thinking about mass stranding or restranding. We wanted an effective alternative to the barbed projectile tags. We wanted to reduce tissue trauma, thereby increasing the attachment duration. And we wanted to be able to consistently deploy tags on the dorsal fin and reducing the risk to the individuals and improving the data throughput to Argos. We also wanted to use off-the-shelf tags. We didn't want to have to develop something new. We'd done a lot of work with wildlife computers to develop a tag that was safe and effective for fin mount purposes. We wanted to be able to use that if possible. And we wanted to try to take advantage of the natural tendency for some dolphins to bow ride and surface right at the bow. So our concept discussions were initiated between the Sarasota Dolphin Research Program and Woods Hole Oceanographic Institution, the vets and engineers in 2014. Excuse me. And then in 2017, we received funding from Dolphin Quest to develop a prototype and begin the design and the testing. Basically, the way the tool works is that there's a tag that's sitting inside the holster. You're looking at the bottom of the holster. The tag is brought up against the trailing edge of the dorsal fin. In the back of the V where the tag is a trigger. When it contacts the dorsal fin, pneumatic pressure then sends a pin through the dorsal fin, securing the tag. The dolphin swims out of the tool, swims away with the tag, beeping. Great concept, great if it works. It took a long time to get to the point where it would. We did a lot of revisions and iterations. We did bench testing on carcasses at Hui. We did field testing in Hawaii with Robin Baird and company. We did field testing offshore of Sarasota. We used video extensively that showed the need for speed. These dolphins have been around for 25 million years. We haven't been. They've developed the ability to respond very quickly to threats. And one of those responses is the ability to be able to roll 90 degrees in 30 milliseconds. And so we had to learn this the hard way by constantly increasing the pressure going through the system to increase the speed. We suspect that that evolutionary pressure for doing that came from shark bites. If you look at the shark bite on the slide here, you see both sides of the peduncle. When the sharks come up and grab the peduncle, they bury their teeth in one side, the dolphin rolls out, leaving the scrapes on the other side. So we then went through more iterations and changed the attachment material to get longer durations. And by 2023, we had our first successful deployment on an Atlantic spotted dolphin off Sarasota. And here is the first deployment on a dolphin named Hannah. And so it happens very quickly, and the dolphin swims off. We've done 7 more deployments on Atlantic spotted dolphins since then. The responses typically involve short-term responses where the animals will swim away quickly and leap. But as you can see in the middle top picture, the animals return to the bow oftentimes, and we're able to get these photographs of the dolphin well seated on the fin as they come back and ride at the bow again. So it doesn't seem too traumatic for the animals. We've been able to compare with Atlantic spotted dolphins the duration of attachment or the duration of transmissions. And what we find is that our median so far with 8 deployments is 38 days with a maximum of 89 days for tags that are designed to transmit up to 3 to 4 months, ideally. The pantropical spotted dolphins that have been tagged by Cascadia Research with limpet tags have a median attachment duration of 13 days. So we're getting 3 times the attachment duration with these fin mount tags through the tadpole, as has been possible with the limpet tags. We find that the movements of the animals are consistent with those that we've tagged through catch and release. These graphs show that the combined catch and release and tadpole deployments of satellite link transmitters on spotted dolphins off Sarasota. The exceptional sightings to the north in the left-hand map were associated with hurricane passage, but the animal did return after that. It actually gave us the opportunity to study what dolphins do during hurricanes, which has not been possible before because my wife won't let me take a boat out. So in June 25, we got a grant from Dolphin Quest to actually take the tadpole someplace and try to apply it. And we went out to Hawaii, worked with Robin Baird and Cascadia Research Collective to tag dolphins off Oahu. And we were successful with tagging 9 Hawaiian spinner dolphins out there. And what we found was very gratifying for some of those of us who have been around for a long time and studied spinner dolphins with much cruder tools a long time ago in the 70s and 80s with Ken Norris. Back then we had VHF tags that we could use just to look at movements, and then we hypothesized what the animals did when they went where they went. And we couldn't really follow them very well in the dark. With the satellite link tags, what we did was confirm the work that Ken initially described 50-some years ago, showing these animals do have a diurnal pattern of feeding. They go offshore at the later part of the afternoon, they go out to deep water, they dive in deeper water, and we were recording depths of up to 300 meters for these animals and durations of more than 7 minutes, which for these little tiny dolphins was a bit of a surprise for me. But we also showed that their dives during the nighttime were much deeper than any of their dives during the day when they come into the shallow bays and rest. We also were able to document movements around the island of Oahu and between islands. We had 2 dolphins that went over to the west side of Molokai, and this is all part of the 4-island, the main island 4-island stock of dolphins as proposed by NOAA for their stock assessment. So they were consistent movements by the animals. And we were able to monitor them simultaneously and look at their movements around the island. Yellow is daytime movements, black is nighttime movements. They move offshore to the shelf edge at night. They come back into the shallow bays in the early hours and spend their days near shore and then move back offshore at night. And the composition of the groups were changing. Sometimes the animals came together, sometimes they separated. It's very fluid. Again, something that was proposed by Norris and company many years ago, but very interesting to be able to demonstrate it with this kind of tagging technique. And so we've now finished tracking the spinners out in Hawaii, and the result is that the median tracking duration was 41 days on one of the most fragile animals that you can imagine for trying to secure a tag. So we're very pleased with the results there. There are a few things we still need to tweak. This is definitely a work in progress. So some considerations. We need to try to perfect the tool and technique. That doesn't mean we can't be using it now as a conservation tool. We've only successfully tagged 2 species of dolphins. It's not necessarily a good fit for all species in all situations. Bottlenose dolphins have yet to be good candidates, but I'm trying to convince them otherwise. To date, it's only been done from 3 different boats, but it's been done from 3 different boats, and we need to try it from a variety of vessels to see how widely it can be used. We can increase the probability of success with bow riding dolphins that surface slowly and predictably in the appropriate position and at the appropriate distance below the bow. We need calm seas with minimum vertical movement of the pulpit, which moves the tagger and the dolphins. We need a stable tagging platform with minimal distance above the water surface. We need long, strong arms, which is why I haven't attached any of the tags yet. Experience is important. We have got a lot of different things moving in multiple dimensions, and putting that all together and doing it just the right time is really important. To date, only 4 people have tagged with it, but 4 people have tagged with it. So if there are potential collaborators who think this might be useful for their situation, they should send us photos of the potential tagging vessel and video of their bow-riding dolphin so we can give an assessment of whether or not it's worth the effort. As a side benefit, we've also been able to determine that this is a conservation tool for other species. While it was in development, colleagues of Michael Moore's at Woods Hole took it out and used it on great white sharks and on whale sharks. So yay, something we hadn't planned on, but something we hope will develop. It is being developed into the future, and something that can make a difference. All this has been written up, the development of it, of the tag deployment tool has been written up in this publication. The QR code is there, or I can get you a copy. So we're very grateful to those who made this possible, Dolphin Quest especially, for getting us off the ground with the funding to make this happen. The National Marine Mammal Foundation worked with us in refining the tool and getting it into the field for some of the final tests, and many others have made a difference for us over time. So thank you very much. But I can't actually see the thing down there. Do I have any time?
We can maybe move the timer somewhere else at a break.
So our next presentation is by Chris Zadra, who will talk to us about UAS-based deployments of suction cup tag attached biologging tags. Bombs away. I added that part, sorry.
All right, good morning, everyone. I have a lot of slides and cool videos, so I'm just going to get right into it. And start with thank you to everyone. This is probably a non-exhaustive list at this point, but obviously we wouldn't have been able to do all of this without having the opportunity to collaborate with all these different groups and all their fieldwork projects. So thank you to all of these people and more. This is normally the last slide, but I just wanted to put at the beginning of this just to give a frame of reference for how much flying around we've been doing the last 3 years. The big numbers there, 235 successful tag deployments in 3 years. Very good success rate. This is not just me. There's a couple other pilots at this point, maybe 2 or 3 dozen tags that have been, we've trained up NOAA pilots and other team members to do deployments. And it's been used on 7 different large whale species, and also a range of different versions of D tags, CATS tags, and ACUSON tags. So when we started kind of end of 2021 developing a system. We wanted to make something that was replicable for other groups. We're all about, designing a tool that we can share across the globe and get this in the hands of other research groups. So everything is, for the most part, 3D printed modifications for off-the-shelf drones. So originally we were using DJI. We've also expanded to the FreeFly Astro now with help from Julian Dale. But all the mounts and the stabilization systems and everything is, again, 3D printed for off-the-shelf drones and also off-the-shelf tags. So we're not doing any designing of new drones or designing of new tags to make this work. It's everything in the middle. The key things we need are a way to stabilize the tag so that it obviously lands suction cups down on a drop, and a way to hold onto the tag and have the drone hold on to the whole system. So you can see there originally it was just kind of a pin release system. From the flying methodology part of it, so this is the kind of third person view. This is dropping the smaller D tags with our original, what we call a lawn dart stabilization system. The challenge with this system was that we needed to be sufficiently high for the tag to have enough time and velocity when it drops for the aerodynamics to make it work. And the higher you are, the smaller your target is as the pilot. So it made it a little bit challenging, but still a very functional system. In contrast with the larger camera tags, the CATS tags, because they were much larger and heavier, that lawn dart would have also needed to be bigger, and it was just too cumbersome. So The methodology here was for the pilot to fly basically as low as we were permitted and rely on the pilot's ability to have the drone as horizontal as possible when you release the tag so that it didn't have enough time to kind of roll out of orientation. But again, also this was a challenge for pilots because when you're that close to an animal, this is a blue whale, so the scale is a bit ridiculous, but you don't have a lot of kind of perception of where you are on the animal. And you need a hefty drone to be able to survive that. So fast forward a little bit to kind of where we are now. At the end of 2024, we developed what we call the gyro. So it's a spinning weighted flywheel. So that large yellow wheel has a bunch of weights in the end of it, and we can spin it up really fast on the drone. And when that's falling in free fall, that physically stabilizes the tag. And what's great about this is it allows the pilot to kind of come in at any altitude, and it also works for a variety of different tags because we can adjust the size and weight of that flywheel. So in the middle there is an Akusan tag on a humpback whale, and on the right is a camera cat's tag on a right whale. And it just keeps the tag perfectly flat every time, which has really drastically increased our success rates. So back in time again to when we first field tested this in Loretto on blue and fin whales, we immediately saw the benefits of a UAS-based system. So this was across 21 successful tag deployments. Our average was we were able to tag animals from almost half a kilometer away, and in under 3 minutes from the time we launched the drone to the time we dropped the tag, with pretty big maximums there. Max and minimums, the maximum of up to a kilometer away and as quickly as 43 seconds from launch to tagging. There's a paper there with the results from this first field test by Dave Wiley in 2023. So after that, we thought about what would the best use cases for a drone-based system be. And kind of the obvious ones are hard to approach species, species that are typically more evasive from vessels, endangered species that you want to minimize impacts on, and projects that might require reduced vessel noise if you're doing acoustic studies or stuff like that. So the first example of that was with sei whales in Massachusetts. Kind of an enigmatic species there that typically people don't bother studying because they're just kind of too elusive. And you can see here with the UAS system, kind of not super bothered by the tag deployment itself, just right into feeding. And the sei whales were typically a challenge for traditional pole tagging because their feeding behaviors here, they would just be inches beneath the surface for 10 to 12 minutes, which meant tracking them in a boat was extremely difficult. But the benefit of the drone is this is obviously the drone's eye view. We can follow them when they're beneath the surface, whereas from a boat, you might not even know that the whale's there. And just waiting for an opportunity for a little bit of back to be exposed and then release the tag and drop. Another example is, or another use case is tagging whales in a group. So this is gray whales in Puget Sound, where with pole tagging in the past, when they would try to tag groups of animals, they would often split up the group when they approach closely with a small boat. It's hard to see here, but you can just about tell that after we tag that animal, the other two already have cat's tags on them. So they're still in a group together and not very much disturbance at all by the drone-based deployment. So at this point, we started working on larger offshore vessels, so not just from small boats. This opens up a whole another area of application for UAS drone deployments. As long as the boats are kind of maneuverable enough still, I forgot to mention this, but you might have noticed that our stabilization systems are releasing from the tags when it impacts the whale. So those are just meant to float in the water and we can recover them and pick them up and reuse them. So the caveat for large offshore vessels is they need to be maneuverable enough to be able to still spot that thing in the water and go scoop it up out of the water so we can reuse it. One project we did was with North Atlantic right whales and kind of Great South Channel area, where it was often a challenge, both weather-wise and logistics-wise, to launch a dedicated tagging boat. So this was the first time we decided to just launch from the big boat. And it's hard to tell from this top-down perspective, but this was in sea states that they didn't want to launch the small tagging vessels, but we were still able to get some tags onto right whales. We've been collaborating a lot with Ed Lyman in the Hawaiian Island Humpback Whale National Marine Sanctuary in Maui. And another use case is approaching entangled animals. So animals that are kind of already in a stressful situation and where you might want to be saving that first close boat approach for your actual disentanglement. We are able to approach these animals. You can see it's a mom, calf, and escort, and after the tag goes on, even like the mom and calf don't even respond to it. So able to collect data that these disentanglement teams might be interested in without negatively impacting their chances of success on potential approach with the boat for a disentanglement effort. And a quick tangent on that, this is something else we're working on right now, taking the success of deploying tools from a drone and trying to apply that to deploying disentanglement tools. So I won't talk too much about it, but again, it's the idea of using drones to replace those first boat approaches to try to reduce impact on the animal and make it so that when you do need to use that first boat approach, that first approach is always like your best chance of success. So we're experimenting with the team, disentanglement team down there in Maui on deploying, extra working lines for them, cutting grapples, telemetry buoys, etc. Oops, quick video there of us dropping a grapple and pulling and cutting some lines. Let's get that. So this was kind of a culmination of all these best use cases, working with Rice's whales last summer in the Gulf. We were working off the Song of the Whale, a large research vessel. We were using our new gyro deployment system, and we're working with an extremely elusive species. And so, yeah, this is kind of how it all comes together to use a drone-based system, dropping a camera tag on a Rice's whale. And I think also probably the smallest species we've worked with so far. So tag data is important for many different reasons. These are some of the more recent topics in some of the field work we've been doing. Field metabolic rates, estimating that on different species. The tags are obviously good for measuring activity, and you can get stroke rates, breath frequencies, both from the accelerometry data, but also off the camera tags. You can often just see at the surface when they're breathing. And we've done a couple different interesting noise exposure studies. All the wind farm construction with the Duke's WOW program. And we've also done some feasibility studies with ABR tags and deploying ABR tags, which is 2 slides from now. So one of the recent things we've been doing is trying to miniaturize this onto smaller drones so we can work with smaller species. Our lovely model Jonathan there is holding a DJI Mavic with the gyro deployment system. But again, we can just pop weights out of that flywheel to make it a lighter system and really miniaturize it down. So this is us using that Mavic to deploy a tag on a humpback whale this summer. But the hope is that this can be used for even more elusive species that might respond to some of the larger drones we're using, or just, yeah, being able to approach smaller species, maybe beaked whales. And then ABR that I mentioned. So an ABR tag has electrodes that you want spaced out. So typically there's a modified D tag in the front that has an electrode in one of the suction cups, and then we have a single suction cup, and it's a kind of double gyro system. You can see the deployment there. So both gyros pop off, and a tag and a single electrode are both remain on the animal, which from a pole tagging thing is very difficult. You may have heard of the success with the minke whale that a team had, I think, in Norway, where they actually had to capture it to put this on. But this ABR hasn't really been used on baleen whales a lot, and we're hoping that this is kind of a proof of concept for a potential deployment method to use on larger species. Oop. Last slide. Uh-oh. Not responding. I think it just crashed. Wait for it to respond.
Oh, interesting.
My last slide is just that we are,
It's not responding, so it's just.
yeah, I'll just have to restart it.
So. Well, fortunately, I think we're ahead of time. You've got 5 minutes.
5 minutes,
So
okay.
those online and in the audience, bear with us.
Can I take some questions, or?
Yeah, absolutely.
Okay, we can do questions. The last slide is that we're open for training and open access. There's a QR code on that last slide for getting access to 3D files if you want to print and make things yourself. And we recently were just awarded a grant to work with NOAA on developing training protocols specifically for NOAA pilots to get this into the hands of more research groups. So yeah, questions?
Very cool stuff, Chris. Thank you very much. Do we have any questions from the commissioners or the scientific advisors? Sue.
Very, very cool presentation. I could watch those videos all day. I'm really curious about your capabilities with recording sound. So you said you have tags that are able to do that you're dropping on whales. Could you say a little bit more about that? And
Yeah.
where you did that?
Yeah, so again, we're not doing any of the tag development. We're using tags that other research groups are using already. The most popular one, I think, is the D tag, which we get our D tags from Alex Shorter at the University of Michigan. But all the tags, for the most part, record acoustics of some form. The camera tags are generally noisier because they're bigger. Yeah.
How many of those have you done?
I don't know the number of D tags versus CATS tags. It's probably pretty split 50-50. Yeah, so over 100 D tag deployments for sure.
Aaron.
Yeah, thank you. That was a very entertaining presentation. And following up on the question of recording sound, I think a lot of the DJI drones, they can record in-air sound as well as video, can they?
No, typically they're just video.
Have you ever explored putting a microphone on here to see if you record the sound of the whale blows? It looks like you can get close enough to be quite within the blow, and I know the Snotbot's been collecting sounds. I know the drones make a lot of noise on their own, so I don't know if it would work, but catching the sound of an exhale would be quite interesting.
Yeah, it would be. You can typically get that on the tags, but yeah, we haven't done it on the drones. And the Snotbot is also just same DJI drone, so we don't actually record sound for that. If there's a video with sound, it's just put over in post. But yeah, there's a question in the back here.
Yeah, I was just wondering if you could talk a little bit more about the accuracy with the location using the plant to put an ADR tag.
Yeah, yeah. So typically on a
Repeat the question for the audience.
oh yeah, okay. The question was accuracy and in regards to the ABR tag. Normally on a non-ABR tag, the objective is just kind of get it on the whale. But we do have some level of accuracy if we want to place it in a specific location. The ABR tag, obviously, you want it over the brain, auditory or brain response, brainstem response. So we did a couple of attempts where, we don't want to push it too much because you don't want to drop it on the blowhole. Very pilot skill dependent. And part of the challenge with that is your camera is not in line with where the front tag is. So you have to take into account that the tag might be 1 or 2 feet ahead of where you're actually looking. It's a challenge, yeah. But that video there was our best deployment out of 3 or 4 attempts, I think. So every time we were able to get the tag on the whale, how close it was to the blowhole was variable. I'd say our accuracy when we do training is we train on a paddleboard with a bunch of ballistic gel on it that is it's 2 feet wide, 4 feet long. So within that area, we can hit that pretty regularly.
Aaron?
Do you think you could use a laser to help as a sighting or a reference point for improving the precision of the deployments?
We've thought about that. So it might have froze again. But we just haven't bothered with it because we don't want to bring it up as a question for permitting, shooting lasers down at whales, visible lasers. What you don't see is on the pilot screen, we normally have like a diagonal X on there. So the center of the X is kind of our targeting. Yeah. Sorry.
Other questions? Don't know if we have any online. Who's monitoring that?
I didn't see any online. And we got 40 seconds, and we got to restart PowerPoint again.
Okay. Thank you very much, Chris.
Appreciate it.
Okay. Once we're back up, our next speaker will be Cynthia Smith, who will be speaking about the development of a minimally invasive blood collection device for cetaceans.
Okay, great. Thank you. Okay, good morning, everyone. It's really a pleasure to be here. So thank you for inviting our organization to share with you the development of this minimally invasive blood collection device. This project has been going on for a while, so I really want to thank the folks that have been committed to the project for several years now. Drs. Forrest Gomez, Kyle Ross, and Ryan Takashita from the National Marine Mammal Foundation, Dr. Lori Swacky at the Commission, Dr. Michael Moore at WHOI, and then the engineering team from TASO, Emily Welch, Bryce Peterson, and Michael Friend. All right, so Dr. Jill Brolick and colleagues recently published a paper that synthesized the extinction risk of cetaceans worldwide, and they stated that 1 in 4 cetacean species was threatened with extinction, which really underscores the urgent needs for us all to be working together, species experts, veterinarians, conservation medicine specialists, and thinking about how we can help fill critical data gaps related to the health status of at-risk cetaceans. Blood sampling, especially coming from a veterinary perspective, but also from a species expert's perspective, is essential for evaluating animal health. And being able to remotely collect blood from a free-swimming cetacean could be a game changer for those charged with assessing individual animal health, population health, and species health. Additionally, there are animal health and welfare benefits to developing minimally invasive options to sample blood from dolphins in professional care, specifically for animals that are less available for voluntary sampling for a variety of reasons, perhaps health status, environmental or social reasons, or when veterinary personnel are less available. And a minimally invasive option would allow animal care personnel to collect samples without them. So in thinking about how to develop a remote minimally invasive blood collection method, we looked to human medicine for inspiration. And in 2011, the founders of TASO created an easy at-home medical device for remote blood collection called the HemoLink. The device used microneedles and microfluidics to draw fluid, or small volumes of capillary whole blood, into the device for diagnostic purposes. So in 2015, we reached out to TASO to see if their blood collection device would work on bottlenose dolphins. And we tested their off-the-shelf device on the dorsal fin and flank of a US Navy dolphin, and we were able to draw a small volume of blood. But we realized it would take an engineering redesign to get the device to really work on cetaceans. So we were hoping for an easy button, and it wasn't quite that, but we did want to see if with engineering we could actually make it so. So in 2020, we secured O&R funds to begin the engineering process. Through the collaboration with ourselves, TASO, WHOI, the Commission, and the U.S. Navy, we set out to develop a minimally invasive blood collection device for dolphins, inspired by TASO's HemoLink, with a long-term goal of developing a device capable of collecting blood with a broad range of free-swimming cetaceans. Our work was primarily conducted in San Diego here with the U.S. Navy's Marine Mammal Program, where our veterinary team provides continuous care. Access to the Navy's dolphins facilitated the clinical device development, which would have been unattainable with wild animals alone. So our first objective was to conduct an iterative sequence of testing and engineering modifications to TASO's HemoLink device with the goal of reliably collecting 200 to 400 microliters of blood. And once we had a working prototype, our second objective was to determine how blood values compared between capillary blood and peripheral blood, which is collected through routine venipuncture during health exams. So through the course of the project, our engineering team produced 22 working prototypes based on a continuous feedback loop from the clinicians to the engineers. The incision depth ranged from 1.5 to 5 millimeters. So I'll just say that again, we're talking millimeters because we're going for capillary blood, which was optimized to really try to reach those capillary beds within the skin. And we also varied the placement and volume of anticoagulant in the device and on the blade. Our final prototype, which we are temporarily referring to a hemotag, but it's not quite a tag yet, had a 5 millimeter incision depth, which is about the width of a grain of rice. And both the device and needle were coated with anticoagulant. In total, we placed 136 prototypes. 84 were placed on Navy dolphins during routine health exams, and 52 were placed on wild dolphins, 44 of those wild Tursiops, and 8 on Inia. All right, so we definitely met several challenges along the way. And one of them was blood clotting. So that was probably our most significant challenge, which resulted in blood flow obstruction and poor yield. So we had to go through some modifications, and we reached success by adding anticoagulant to the blade and to the blood collection channel. We also struggled with yield, which we managed with varying the incision depth. Warming the dolphin skin prior also helped, and that is something we're still working on. How do we incorporate warming elements into the actual device? So our final device configuration can be seen in the top schematic, which shows the side view of the prototype. The device adheres to the dolphin skin via a pressure-sensitive adhesive film, and when that button, which does look like an easy button, is pressed, a spring-loaded lancet punctures the skin and creates an incision. When the button is released, the blade retracts and an internal vacuum is created to help draw the capillary blood into the device, and then blood flows through the microfluidic channels and into a lithium heparin-coated tube, which you can see in the video. The device is peeled off the skin, the tube is twisted off and capped, and the blood is processed. So once we were having success in San Diego, we tried the device out in the field during catch and release health assessments of wild dolphins in Barataria Bay, Louisiana. This project supported an intern, Candace Remo, from Savannah State through a collaboration with Tara Cox's O&R-funded Diversity, Equity, and Inclusion internship program. And this is Candace here placing a hemo tag on a wild Tursiops. So an additional opportunity for device placement emerged during health assessments conducted in the Amana Reserve in Brazil on Amazon River dolphins. So Dr. Forrest Gomez also placed 8 tags during this effort on wild Inia. And let's see. Yes, all right, so moving on to our next objective. Once we had the reliable prototype working, we moved on to our second objective, which was to compare blood analytes between capillary blood, with the prototype device and traditional venipuncture methods. So for the comparison study between the two, we collected paired samples from 29 dolphins with a broad range of age and size and a mean blubber thickness of about 2 centimeters. In all samples, a PCV and total protein was measured, and a blood smear was made for a manual total white blood cell count and differential. In 24 of the 29 paired samples, we also ran iStat Chem 8 Plus cartridges for electrolytes, TCO2, glucose, BUN, and creatinine. So for several of the variables, we found no statistical significance between the prototype capillary blood and fluke blood values, and that's what we were looking for there not to be a difference between the two. And we did this using several different methods of statistical comparison. So the hemotag did provide a really good representation of PCV, total protein, total white blood cells, monocytes, eosinophils, glucose, and creatinine, but there were other variables listed where we did find a significant difference between the capillary and PAVR blood. And this might simply be because there are differences between capillary and PAVR blood. So we might have different reference ranges that need to be developed for capillary blood. But for the ones that I listed first, those are looking like the same reference ranges as with peripheral blood. All right, so here's a couple of examples of comparisons between the two PAVR blood and capillary blood, and periarterial venous reedy blood, which is where when we take samples from the fluke blades, that's what we are sampling, and the capillary blood. So in the box plots of PCV on the left and white blood cells on the right, the black line through the middle represents the median values, and the blue lines are the upper and lower reference range values. So you can see strong agreement between the two collection methods. For PCV and for total white blood cells, there was a slight increase in the median value of the hemotag capillary blood values, but no significant difference. So in summary, we developed a minimally invasive blood collection device prototype for cetaceans that is currently reliably collecting about 200 microliters of blood, capillary blood. The comparison of capillary blood from the prototype device to PAVR fluke blood showed concordance for several clinically relevant values to include PCV, total white blood cells, and total protein. And we successfully collected blood from two species, the bottlenose dolphin and the Amazon river dolphin. We have submitted a follow-on proposal for the next phase of device development, which includes field testing, waterproofing, skin warming, and then adaptation to whales. So I just want to end by thanking again everyone that's been really committed to this project along the way. Randy Wells has also allowed us to bring them to Sarasota and do some testing as well there. And so it's just been a wonderfully collaborative project, and we are really hoping to continue moving it forward so that we can get to the end goal of a remote blood collection device from free-swimming small and large cetaceans. So if you have any questions, please feel free to reach out to myself, Dr. Gomez, or Dr. Ross. We'd be very happy to take your questions and ideas for continuing to develop the device further. Thank you.
Thank you, Cynthia. It's so great to see how this has progressed. You guys have been making great strides.
Thanks,
We do have time for questions, first from the commissioners and the scientific advisors. Francis?
Thank you, Cynthia. That's super cool and really exciting. I'm curious about the need to press the button, which therefore implies that you actually have to hold it, and therefore you're holding the animal. Therefore, you could get a blood sample. So are there plans to have some sort of remote method for pressing the...
Yes, yes. So right now, you're right. You have to depress it and then hands off.
Right.
It stays depressed because that triggers the vacuum.
Yeah.
So the plan is to have that happen. I mean, the grand plan is that we would be dropping it from a drone. We'll be talking with Ocean Alliance about that.
Yeah.
And that it would have a self deployment mechanism, so that that button would be, once it reached contact with the animal's skin, that would be deployed. And then we have all kinds of ideas about sampling at depth. Maybe there's a delay so that once the animal dives and it gets to a certain pressure differential, would then it take the sample? So there's a lot of sophisticated engineering challenges that we're dreaming up. But yes, that is a really important point, is that right now it's a hands-on device, and then the idea is to get it to be a remotely controlled device so that it would do a lot of things on its own.
We have time for one more question. Okay.
Can only one sample be collected per deployment, or is there an opportunity to collect more than one?
Yeah. That's a great question. So right now we're talking about making it bigger for whales. So our next phase is going to be to scale up the device and make the volume of blood that it can collect larger. But I don't think we've thought about having more than one port so that you could be collecting into two different basically ports, and maybe they have different anticoagulants in them. That's an interesting thought. I don't know if that's what you meant, but currently the current design is for one sample, and then you would need to place a second one for another sample. But we're gonna take that idea and play with it.
Thank you so much, Cynthia.
Okay, Peter, no, yes?
Quick question, it's just physiologically, the capillaries on the surface of a whale are always been a subject of myth and study. Where exactly is the blood reaching the surface? So obviously that's something you have to think about.
Yes, and Michael
Are
Moore.
there places that just there isn't going to be any blood?
That's right. And Michael Moore is helping us with that. And so part of the next phase is also to be working at Hui with carcasses and really searching for where is there going to be an ideal location that we should be targeting, or is there enough distribution across much of the whale for us to have success? But we're going to have to figure that out. Great point. Okay, thanks.
Okay, sorry, Peter. Our next speaker is Michelle Shiro, who will be speaking to us remotely about harnessing emerging technologies for a non-invasive approach to marine mammal energetics. Hi, Michelle.
Hi, so I'm going to try to turn everything on. Can you guys see the screen and hear me okay?
Yes,
Perfect.
although we see your side slides as well, not just the presentation. There you go.
Yeah, here we go.
Yep, good.
Okay. Get this set up well. Seems to be a little weird.
We can
Okay.
see. Okay.
Perfect. Okay, so throughout my research, I really tried to use my background in physiology to develop non-invasive methods for assessing aspects of animal health that are challenging to determine using traditional or hands-on techniques. So that's what I'm going to be talking about today, using emerging technologies to get at marine mammal energetics. And that's because an animal's metabolic rate or energetic expenditure both impacts and is impacted by interactions within its environment. But techniques for obtaining measurements of energy dynamics or metabolic rate tend to be pretty invasive, logistically difficult, and costly. From respirometry to having to hoist animals for mass measurements and isotope dilution techniques, with many of these methods often requiring long handling times or that the animals are sedated. So obtaining these measures beyond just a few individual animals starts to become pretty unwieldy. But use of non-invasive methods, and in particular imagery that could be acquired quickly, would allow us to collect indices of energetic expenditures from a lot more animals to better understand how organismal level processes translate to energy dynamics population-wide. And so my lab's been using thermal imagery to measure animal vital signs like respiration and heart rate that are common proxies of metabolic rate. And this actually started during COVID. Like a lot of folks, all of our fieldwork was canceled during the pandemic. And so instead of going to the field, we went to the zoo. And in humans and other species, mainly species without a lot of hair, you can use your general red, green, blue videos to detect heart rates through changes in skin color. Um, But we wanted to see if you could do this consistently in animals with a lot of fur that would obscure your ability to visualize the skin or blubber, and whether you could see respiration and heart rates by way of changes in blood flow with the thermal camera. And so we did this study at the Cincinnati Zoo, and we imaged more than 50 species there. Most of them were willing volunteers who just really liked treats, like this red panda. Others were animals like this cheetah that were anesthetized for their annual checkup at the zoo. And this is my graduate student, Caroline Russillo, taking the photographs, and she's the one who led this study. And so we worked to pull out these pretty subtle physiological signals. And so as an example, this is a gray seal volunteer from the zoo. And as he was a volunteer, he wanted to be upside down. And so just to orient you, this is his eye, this is his nose. And as you can see from this thermal video, there's not a lot to see from this thermal video. We don't see a lot of pulsation. There's no clear signal being picked up. But then we went through and we applied Eulerian video magnification. It's a type of spatial and temporal decomposition analysis to really magnify any changes in color that were existing in the video and at physiologically relevant frequencies. Um, so here you can see pulsation. There's this clear change in the signal through time. And after a Fourier transformation, we can pick out the dominant frequency of the signal. And we found the most prominent signal coming through after amplifying the changes in temperature were always pretty close to respiration rate, which we confirmed by monitoring the animal's rib cage expansion or movement of the nostrils. But there's still a lot of noise that you can see in both of these figures. And if we focus in on this area with the secondary frequencies, the dominant peak here was always very closely related to heart rate, which we confirmed using an EKG or a stethoscope. So this method was successful in measuring respiration and heart rate in a lot of the mammalian species that we imaged, including the seals. And then for animals that we were not able to measure respiration or heart rates, really importantly, we weren't pulling out false signals. So if we got a signal, it tended to be pretty accurate. And we saw relatively low errors between the thermal imagery derived vital sign rates and the true values. So since then, we've started taking these methods out to the field for validation with sedated animals, ensuring the measurements are repeatable and trying to determine how far away we can be from the animals to still take measurements of respiration and heart rates. And so during our first attempts with this and the scientific cameras from FLIR, we were able to detect vital signs from about 5 to 8 meters away from the animals. We would like to be able to do it from even further than that. And so once we were able to get a longer focal length thermal lens, we were able to increase that distance a lot more. So this was a 142 millimeter lens. We're still working to make this a little more practical in the field, but we have some preliminary successful images measuring heart rates in seal pups from almost 100 feet away. So that's me all the way in the distance there, imaging this little gray seal pup. And so increasing the distance would mean we'd really be able to survey the animals from far enough away to ensure we're not disturbing them and are also getting true resting heart rates when we start surveying. Taking this to the field also meant that our environmental conditions were a lot more variable than at the zoo. So we had to modify our processing a little bit because we started to see interference under windy conditions. In this video, you can see the heat being drawn away from this animal with each little gust of wind. And so our first thermal derived respiration rates in the field had really poor accuracy. So that's this figure here. And for respiration rate, we had always been amplifying signals present across a pretty wide range. And so having a wide passband just means that you have a lot of signal noise. And so our signal was getting drowned out by that noise from the wind. But if we narrowed the passband, we reduced the amount of noise, and this actually got rid of that problem almost entirely. So this was something that we now know we need to control for that can be mitigated during processing to make sure that your thermal imaging derived values are still very close to the true values. And that's because ultimately we want to be able to better apply these techniques to free living animals, for example, to track energetic expenditures across lactation or between female pup pairs with measurements being made on multiple animals, even from the same video. I've also been working with engineers at Hui to modify heart rate loggers that were intended for humans to use on seal pups that can just be attached to the fur. The most prior heart rate loggers used in seals required subcutaneous implantation under the skin, or there was some prior work with some similar tags a number of years ago, but they were telemetered. And so someone had to be sitting nearby at a receiving station to get the data. And you could only get the data as long as someone was willing to sit there. But these can log continuously for 9 days. We just trialed them this last year on just a couple pups because we weren't sure how well this would work. But we ended up with these beautiful EKG traces with heart rates for the pups. And then the tag also had an integrated accelerometer. And so we weren't expecting this, but we were actually able to see the expansion of the rib cage. So we could even get breathing rates from those instruments too. So this upcoming year, we'll be able to validate the use of the thermal imaging in free-ranging unsedated animals by having some of them wear these heart rate loggers and also ask bigger questions using these instruments about relationships between the efficiency that pups assimilate energy, how well they can conserve energy during their post-weaning fast, and development of breath holds and apneas. Um, and so ultimately, these changes in metabolic rate should translate to changes in the animal's mass and condition. And so that's also been a really big focus of what my lab has been doing. And again, it's not surprising that there is also a long history trying to estimate animal size using simpler metrics, such as from photographs using body ratios or 3D modeling animals to get a lot of more dimensional information from 3D than you would from 2D. And so this has also been done for a variety of different taxa. But in marine mammals in particular, their extreme mass dynamics is something that's really easily visualized. And so photogrammetric approaches have become very popular. And we see more and more of these efforts taken to the air, like we've seen in many of the talks today. With the rapid advancements, miniaturization, and growing consumer demand for unoccupied aerial systems or drones, this has really revolutionized the ways that we can measure marine mammal condition. The for cetaceans, especially where you're working from a boat, you don't know exactly where your whale is going to surface. Opportunities for photographs are going to be limited because that whale is only going to briefly stay at the surface. And then you also can't see the sides of the animals that are underwater. Often a single meter or top-down photograph that can be taken quickly is going to be ideal. There's impressive freely available programs that will take a suite of morphometrics all up and down the animal's body in 5% increments, taking lengths and widths at each location. And then you can decide if you want to choose to focus on portions of the body that are likely to fluctuate more with changes in nutrient availability, like the animal's core regions. The pinnipeds typically provide a different story. They very predictably haul out at certain times of the year. They often remain pretty sedentary and they haul out for long durations fasting. So there's often opportunities to do a lot more with your imaging efforts. And so we wanted to extend those aerial photogrammetry efforts to 3D and to be able to measure larger groups of animals at once. And so for this work, we went to Saddle Island off the coast of Nova Scotia. There's been a lot of interest in the energy dynamics of gray seals as their populations have increased at this exponential rate all up and down the North Atlantic. And then we did this project with Mike Hamill's group out of Canada DFO and also Dave Johnston's group out of Duke. In the 3D model groups of animals, we started by picking a group and flying 360 degrees around, taking sequential photographs in what's called a structure for motion approach. We flew at altitudes between 20 to 40 meters. And we found that it was really important for us to be taking oblique angle photographs to really capture the low profile sides of the animals. And so importantly, in this study, none of the animals were sedated for the flight imagery. We wanted to test that wild free-living seals would in fact stay still long enough to perform the 3D photogrammetry. And then because the drone knows its GPS coordinates and its altitude, we can easily add a sense of scale to our 3D models. And we did all of our project processing in Pix4D Mapper to create our 3D models. We required that any feature or point be seen in at least 5 of the photographs in order to be incorporated into the 3D model. And often it was seen in a whole lot more, as you can see here. This just helped make sure that the model was less sensitive to small animal movements. And so here's an example of a 3D model point cloud from one of the larger groups that I imaged. And as well as an active breeding colony. So you can see a little of what we call ghosting here. And that's usually the males running around. But more than 80% of the animals imaged remain still enough to get complete 3D renderings. And we made sure the animals weren't lifting their heads or showing any behavioral signs of disturbance towards the drones. So we were able to image whole groups of animals at once. And then in stitching the images together, we created an orthomosaic and then a digital surface model that shows relative elevation. And we can see all the seals sticking out here. And this difference in elevation, that is the seal, is ultimately what we're measuring when we take our volumetric measurements. And so we were able to measure and weigh some of those animals after we imaged them to see that the volume measurements we were getting from the air were very similar to what we were getting from the drone, and that we could estimate animal mass with little error. Mean errors for both the adults and the pups are about 4 kilos. And then extending to some broader ecological questions, we did the study across 2 study years, and we ended up with very 2 different looking years in terms of what the environment looked like. So you can see in this aerial shot, this is the exact same spot at the colony in 2019, and there's ice right around this area. And you can see the colony is pretty sparse. Compared to this exact same location in 2020, there's open water next to the island, and there's a ton more animals densely aggregated. In 2019, the Gulf of St. Lawrence had 90 to 100% ice cover over the month of January. But in 2020, there was only 10 to 30% cover across the month. So we did a couple of fixed wing drone surveys that took thermal images so you can more easily see the animals on this peninsula here. And in 2019, there's all this ice around, and the seals were able to spread out a bit for pupping using the ice as well. Whereas in 2020, this is all open water. And so all the seals had to come back to that land to haul out and give birth because there was no ice around. And so for the animals that we weighed, we also gave them a dye marking so that we were able to find them again multiple times throughout the season for imaging. And we did weigh a subset of the female pup pairs multiple times so that we could ensure the method had the necessary sensitivity to track changes in energy dynamics in our handled animals, as well as applying that to about 700 additional animals that we imaged. And so across lactation, female body volume and therefore estimated mass declined across lactation while pup body volume increased. But the females lost and pups gained volume and mass more rapidly in 2019 than in the 2020 low ice year. And that effect was a little greater in pups than in adults, resulting in higher volume or estimated mass transfer efficiencies in 2019 than in 2020. So that is, even though the females were losing volume and mass, not as much of that was apparent in the mass gained by the pups. So presumably being in high densities would lead to a lot more interactions between conspecifics. So females may have been expending more energy, maintaining their little area at the colony, and a lower proportion of their energy may have been transferred to the pup. But basically, there may just be physiological consequences of shifting from ice to land for breeding. These were also all unmarked animals, so it'd be really neat to look at this in more detail in the marked population or using AI in the future to keep track of individuals by their modeling patterns. We've also been looking at how these methods could be tailored for different needs. I took the drone down to the Antarctic once with Kim Getz while she had been at NIWA working on little seals. And it was March there, so it was very, very cold. But the biggest issue we saw was that now we had a very, very homogeneous substrate that the animals were on, sea ice. And it seemed like the models were taking these erroneous key points and trying to put them together in 3D and ended up with all this artifact in the air that made it very hard to measure any of the animals. But if we change the flight plan to take photographs with the camera pitched at different multiple oblique angles, this really fixed that problem. And so the sail is lying on an insulet pad, but you can see there's no snow artifacts strewn into the air anymore. We also tried some transect flights, since that would allow you to cover big swaths of ground. And again, the oblique images were just really important. If you only took nadir photographs, like most drones will automatically do when you program transect flight missions, the entire sides of the animals are totally missing in the 3D models. But if you could alternate taking oblique and nadir photographs, that really dramatically improved the 3D reconstructions. Just wrapping this all up, we've been really trying to use non-invasive means of acquiring indices of energy dynamics and building this up from the ground to the air very slowly using validations to each step. And ultimately, we just want to be able to get a wider area assessment of animal health within populations. And I think that, imaging will facilitate us being able to capture indices of animal health for far more animals than would ever be possible from the ground. Um, and we're also really interested in tailoring this and using it in different species and species of concern. So if anybody is interested in trying this in a new system, very happy to help. With that, I can take any questions.
Thank you, Michelle. Very impressive quantity, quality, and diversity of work that you presented. We've got a couple of minutes to take questions. Let's start with the commissioners and the scientific advisors. Jason.
Hi, Michelle. Excellent talk as always from you. I'm curious with the thermal camera for heart rate and breathing rate. It seems like from the images you focus on their face, and I assume that you've tried other parts of the body, but you can kind of see even heart rate sometimes along the flank, along behind a foreflipper. Does that just not work with the thermal camera?
Yeah, so we definitely tried a bunch of different locations and across all the species at the zoo that we were imaging, definitely the face or in between the rear legs, if you could see that, was the most reliable place to image to get a heart rate. And then also in the field, we did image because you can clearly just see a pulsing heart rate if you look at the blubber jiggling in the seals right behind the foreflipper. But that's where the blubber is also the thickest. And so you don't see it with a thermal camera. So right around the face was really reliable.
Francis.
Thank you for all that, Michelle. Another question on the thermal camera. Do you think there's enough sensitivity to pick up febrile animals? If we're thinking of screening pinnipeds in the field for avian influenza, could there be a heat signature in the mucosa or the eye?
I think that would be more difficult. So for the vital sign measurements, we're not actually interested in absolute temperature. It's just the change in temperature. And so with thermal cameras, as many of you probably know, it's just really sensitive. So if there's shade or whether the animals are molting, you get different thermal signatures from the fur than if they're not. And so if you wanted an absolute temperature, that might be more difficult. Yeah.
One more quick question. Yes, Aaron.
Hey, Michelle, just a question about the thermal cameras. What's their frame rate, or how many samples per second can you take?
It's 30 hertz.
All right. Thank you very much for a very great talk. Our next speaker is also remote, and this will be Ryan Kelly speaking to us about advances in eDNA monitoring of marine mammals, current capabilities, and future directions.
Okay, let's see if I can get set up here with the with PowerPoint. And thank you for having me. Not seeing my PowerPoint. There. Okay. Can I get a confirmation that you can see what I see?
Yes, we can see what you see.
Great. Thank you very much. And thanks for having me. That is an incredibly impressive talk to have to follow. So I will do my very best here. I am pinch hitting for Mike Weiss, which are big shoes to fill, but I'll do my very best to talk about some work we've been doing with environmental DNA and it just sort of in general, and then also specifically as applies to marine mammals and cetaceans in particular, because that is the interest of ONR, who's funding much of this work. So as a big picture, first principles here, eDNA is just environmental DNA that's genetic traces recovered from the water. So the process by which we're getting these signals, as many of you know, it's not very high tech in terms of collection. We are literally getting, bottles of water, either surface or depth, and then pulling the DNA out of it. We can sequence that DNA, otherwise treat it, and find out what's there. So that technology has been around for a while. And it continues to amaze me that we can get things, we can get a wide variety of cetaceans from water samples. That signal should be fairly rare. We see it reasonably commonly, and that's really cool. But is it useful? We know already that there are fin whales in the world. And so it's not surprising that there is fin whale DNA in the world. But how do we make use of that information? What are we really sampling, and how do we turn it to something immediately useful for us? So this is how we think about the world, that that the organisms we care about here are multicellular. Those organisms are made up of cells, and those cells have DNA in them. All of them do. And so when you have more of a species in the world, you must have more of its DNA in the world, full stop. So that's useful as background to have in your head, that when there's more of a species, there must be more of its DNA. And then you layer that on top of the idea that organisms are discrete. So picture a whale swimming around the world. That whale is in one place in time, at a given place in time, and that it's shedding DNA out into the world. That DNA is subject to, so it's been shed, it's also subject to these other forces it's constantly decaying, and it's being transported around. And so what we observe when we observe eDNA in the world is not just it's not the same thing as observing visually or acoustically the organism itself. We're observing this sort of integrated product of shedding and decay and transport and maybe some other factors. We're observing the integration of that. And so what that means is that the eDNA signal from a given species in the world is smoother than the discrete animals themselves. So, okay, with that as prelude, what kinds of information can we get out of genetic residual material in the world, and what might we do with it, given now that we understand that it's out there, that more of a species means more of its DNA. Also, we do not expect it to overlap precisely in space and time with the discrete animals that might be giving off that genetic information. So a lot of people think about detection. First of all, do we see the thing's DNA? Do we not see it? That's useful. Yes, that works. We can get presence, and then when there's more DNA, you're more likely to detect it. So it does give you some quantitative signal, but it's not very much. We can also get information about the concentration of the particular species DNA in the world, or RNA in the world. That gives us a lot more quantitative information about maybe the abundance of species, or something about how close in space or time it was. And I'll show a slide coming up with various fragment sizes of DNA and RNA, and those have different degradation rates, which help us understand a molecular clock by which we can date the parcels of water, or the cells that we're seeing in our samples. And then finally, sort of a frontier here is the sequence information itself. We can get haplotype information from the DNA, and that, by population genetics theory, right, that relates to the size of the number of contributors to the sample, or the size of the population. Functional genetics, like what genes are expressed, and then a real frontier, methylation, we can, tells us something about the age or the physiological state of the animal that has shed the DNA. All of this is available from water samples. And I should say, all of this is available from water samples that you didn't necessarily take for this specific purpose. And so I'll show in just a second, we're actually working closely with NOAA and reusing samples that were taken for a different purpose, repurposing those to look at cetaceans. So again, the challenge it's not the question of, is there this information in the water samples? There is. Now, how do we link it to the parameters of interest in the real world? And I should say, I made mention that, that ONR is supporting this work. We're doing this in collaboration with NOAA, with Scripps and Cal Coffey and Bryce Simmons down there, right where you're sitting in that room, and also Len Thomas at University of St. Andrews. So one question always that's going to come up is about DNA in space and time. How quickly does it degrade? Where does it go? Where is it shed? And here we've got a lot of advantages. We're working in the Hood Canal in Washington State. There's the Navy base that has a captive population of Tursiops, of Atlantic bottlenose dolphins. And we've got a lot of information about the oceanography of that area. In particular, we've got this very high resolution downsampled oceanographic model with 10-meter resolution. So we know where the water's going. And the cool thing about the Atlantic bottlenose dolphins is they shouldn't be in the Pacific, right? This is not a native species. And so anytime we see Atlantic bottlenose dolphin DNA anywhere close, we know exactly where it came from. So we know the source of the DNA. We know where the water is moving. What this allows us to do is do particle tracking. We can forecast where the water's going. We know where the DNA is coming from. We can figure out, we can make predictions about where it goes and when, and then we can go out in a boat and see if our model's correct, and we can adjust accordingly. And this is Jillian Zhang's work, was published earlier this year, showing that basically where we expect high concentrations of DNA, we are seeing that, and so that works reasonably well, and that's a forecast. And then we combining this with new information. This is Pedro Brandau's work that was just published a month or two ago, with the, as I mentioned, the different fragment sizes of DNA and RNA, they're decaying at different rates. And what that means is when we grab a bottle of water, it contains a mixture of all these different genetic signals, and we can actually triangulate sort of the age, we think it's the average age of cells in that bottle of water, given the different fragments that are present. And so we can work backwards in time, ask how old, when was this shed on average? And then we're looking to combine that with the information from the oceanography, the knowledge about the system, to do hindcasting, to say, hey, given a detection of DNA or RNA in the world, when and where was that animal? And so at scales of, this is fairly small scales of 1 to 10 kilometers, this is again the Hood Canal, we can trace that DNA dispersal quite well, and we can start to date these things and hindcast backwards in time, which would be pretty cool. And we are doing that, over the next few months or so, working to integrate all this stuff into a single model. So then at a bigger scale, I mentioned the reuse of existing samples. This is the west coast of the US. Obviously, as you know, this is about 10 degrees of latitude between the Canadian border and San Francisco. And these, in the facets, these are depth bins from the surface down to 500 meters. And these samples were taken in a big grid by NOAA for fisheries purposes. In particular, they were looking for hake, and these are samples taken from 2019, but we've got samples from every other year. They do this research cruise. So we were able to reuse those samples, query them for vertebrates in general, and not just fish, but vertebrates in general, and see cetaceans. We see cetaceans in about half of those samples. So those samples are in the freezer. We can query them over and over again for different, looking for different things. And in this case, we're able to see whatever that is, 18 or 20 different cetacean species of interest. And we can see them at, what's at different depth bins. We can infer things about their ecology. Again, for the folks in that room, none of that is a surprise. Like, the dolphins are going to be at the surface. That's not interesting or surprising to you. But what is interesting and surprising is being able to take all of these species from, the same sample potentially, look at concentrations of those DNA, and then map that over space and potentially over time. So that's what we'd like to do. And here we're learning from fisheries, working with our NOAA partners. These are those same samples in the context of fish. We've been able to use those same samples and say, okay, here's how the sardine DNA is distributed over space and in 3 dimensions here. Sardines are predominantly the surface. That's anchovy, sardine a little bit further south, but a similar pattern. And you see different species at different depths and so on, create this heat map. Where the species is where the DNA of that species is. And I would assert that, in those quantities, there's a proportionality there. Now, what that proportionality is that slope of that line between number of individuals or biomass and concentration of DNA is going to differ by species and by context. And that's a conversation we can take up maybe offline or in the questions. But this is what we're now working towards over the next, 6 months or a year. We're doing this kind of smoothing and quantitative relationship of the DNA for cetaceans. It's easier for fish because fish are more continuously distributed over space. And as you saw, the cetaceans can be a little bit, more stochastic in their detections. But this kind of map is now what we're working on with our NOAA partners for cetaceans along the West Coast. And then finally, the last step here is a quantitative model that will be familiar to many of you in terms of stock assessment, getting to quantity. And there, what we need is multiple streams of data that are coming in to be able to talk to each other. And this is where the work comes in with Cal Coffey and Scripps and Len Thomas in St. Andrews, to be able to join DNA with other kinds of observations, acoustics, visual, and say jointly estimate the density of species of interest. And so here again, learning from fisheries, this is Gled Guri's work that came out last year, did exactly that with fish, and we're going to use a version of a similar model to do this, moving from DNA and joining it with other streams of data to be able to make out of sample predictions about abundance. And in this case, Gled was able to use 3 years worth of trawl data joined with molecular data and can successfully predict the fish in the net in a trawl out of sample year using molecules alone, having trained the model in 2 years, predict the 3rd year, and it works quite well. So that's what we're working on doing with the cetaceans in a joint model going forward in the next couple of months to year. And I'm going to stop there because I'm out of time, but boy, is there a lot to talk about and a lot of people to thank. So I'll take questions, and thank you very much.
Thank you. That was really great. We do have a couple of minutes for questions, and do I see Randy Reeves with a question? I see Randy Reeves.
Yes, you do, believe it or not. Ryan, you put me in mind of it's strange, maybe esoteric question, but demonstrating extinction with eDNA of something like the Yangtze River dolphin and maybe the vaquita. I mean, how long does the signal remain in the area or in the world, for that matter? At what rate does it disappear with the creature itself?
Sure. The second part of that question is easy to answer, and the first part is maybe impossible. The second part of the decay rate, I showed that slide, and we've got a lot of examples of this, but the half-life of DNA in ambient seawater is going to depend a little bit on temperature, but basically you're looking at hours to days. You're not looking at weeks. So if you see the DNA, that animal was there close in space and time. Within hours and within hundreds of meters probably. Proving a negative is sort of philosophically difficult to impossible. And so how do you? I'm not sure I would be able to point you in the direction of being able to establish definitively absence or extinction, but rather establishing presence is easy.
Thanks.
Thank you very much, Ryan. We're out of time for more questions, but we will have the panel discussion after the break. Our next speaker this morning is Ashley Barrett-Clough, who will speak about what can epigenetics tell us about aging and health of marine mammals.
Perfect, thank you. Thanks, Randy, for the introduction, and thank you to the Commission for inviting us here today. It's a real pleasure to be able to get to share this work with you all. So keeping in mind the focus of the stock assessments, why do we need to know the age of marine mammals? Why is it actually important? Well, I think there's multiple reasons. The main ones are that it does aid our interpretation of biological information. So for example, if we're looking at blood sample results, we might have different reference ranges for a calf versus a geriatric animal. It could also give us a more complete understanding of the population reproductive capacity, so how many females are actually of age of sexual maturity and are able to positively contribute to an increase in population size. And it does also provide us with an overview of the population demographics. So do we actually have an aging population, or do we have a skewed population where there are higher numbers of young individuals? So for the focuses of this talk, I'm going to primarily use the bottlenose dolphin as an example, but I do just want to highlight that there is application to many different species of marine mammals for epigenetics in general. So in bottlenose dolphins, prior to epigenetics, age was previously estimated through a hands-on tooth extraction and cross-section of that tooth and counting of the growth layer groups. We were really interested in being able to estimate the age of an animal remotely. So again, with the stock assessment perspective, it would be really interesting to be able to obtain the age of a dolphin through, for example, a remote biopsy sample. So this was where we started to explore epigenetics. So for those of you that are not familiar with epigenetics, it's back to biochemistry 101, I'm afraid, but it's relatively straightforward in that we are looking at the changes on top of or outside of the DNA. And specifically, the type of epigenetics that we're interested in is DNA methylation. So that's the addition of a methyl group, which is the CH3 there, to a specific area of the DNA. So if you remember your base pairs from your biology days, it's to the CPG site that we're interested in. And the addition or loss of a methyl group to that site is what allows us to be able to determine how old an animal is. So this basically has come to light in the form of an epigenetic clock. So the degree of methylation can be determined over time for specific species, and this can allow us to understand an estimate of how old the animal is. So in bottlenose dolphins, we decided to focus on skin samples to be able to perform this methodology remotely. And a skin sample was taken through remote biopsy, or in most of our cases through a hands-on physical health assessment. And the DNA was extracted from this skin sample, and then we looked at the degree of methylation over time in these samples. So we collaborated with the Clock Foundation, and we used an array that had 37,000 different CPG sites and looked at the degree of methylation in these sites. So historically, the use of epigenetics to estimate age was first established in people back in 2010. So it is a relatively novel field in the world of marine mammals. It was first applied to a marine mammal in 2014, to the humpback whale. And more recently, you can see that there has been a great increase in the application of epigenetics to marine mammals just in the last 5 years. What we found from this research is that having a species-specific and tissue-specific clock will provide you with the most accurate age estimation. So if you think about it, different tissues age at different rates. So having a consistent tissue in the samples will provide you with a more accurate approach. Where this can be challenging is for the endangered species. So how do we obtain sufficient known age samples from an endangered species to be able to calibrate the clock in the first place? And this is where clocks of closely related species, or even multiple species having, such as an odontocetes clock, might actually be more applicable. So spoiler alert, it's very accurate, which was very exciting. So the work that we have been undertaking out of the 37,000 CPG sites, the epigenetic clock in the skin of bottlenose dolphins actually selected 211 CPG sites to estimate the age. And it actually came to an estimate of less than 2 years across the full lifespan. So we included dolphins in this study that were up to 58 years of age. From a stock perspective, we were able to look at samples from multiple different bottlenose dolphin populations. We used samples from the US Navy Marine Mammal Program here in San Diego to have exact known age dolphins, as well as from known life history information from the long-term studies in Sarasota Bay, Florida, and through Charleston, South Carolina as well. We also had known age samples or estimated age samples from tooth samples from varying different locations. So over the last several years, we have been able to gather an impressive 1,000 samples at this point, and to be able to estimate the age. And so you can see with the increased sample size, we've actually been able to reduce the median absolute error there down to just 1.6 years across the full lifespan. So one of the challenges, I think, with the epigenetic clock is this discrimination between chronological and biological aging. So biological aging is something that you've probably seen a lot about in the news. It's thrown around very, very commonly. Everybody wants to look younger. Everybody wants to feel younger. And so I think there's a huge drive for research in this in the human field. And so basically, the concept is that you can have a known chronological age, which is obviously your date of birth, your actual age since birth. But you could also have a biological age, which is a bit more of an indication of how healthy you are. It could be representative of your lifestyle. And so to use this graph as an example, you can't quite see my cursor there, but basically if we look at the 70-year-old, for 70 years old, you could be very fit and healthy, like the guy that's waving his walking stick in the air and doesn't need that. And we all want to be below that line. So we all ideally want to have a biological age that is lower than our chronological age. However, due to lifestyle factors or even genetics, we might actually see our biological age being higher than our chronological age. And this is termed age acceleration. And so none of us really want to be the person that is bent over at the top of the graph there and is actually, for a 70-year-old, appearing to be a lot older. So how does this apply to marine mammals, and why are we interested in this? Well, we're interested in it from a population consequences of multiple stresses perspective. And so if we have a population that is experiencing lots of different stresses, is it possible that we would see an increase in their age acceleration or in their biological age? So one of the populations that was involved in our study was the dolphin population in Barataria Bay, Louisiana, that had unfortunately been exposed to the Deepwater Horizon oil spill. So we were able to have estimated or known ages through life history information, through doing health assessments in this area over multiple years, and looked at the DNA predicted ages from this cohort. And what we found was that it was more likely that these dolphins were going to be demonstrating age acceleration than dolphins in other non-oiled areas, such as Sarasota Bay in Florida. So we're not at the point yet where we're able to dial this in to say specifically that the oil was what would be causing age acceleration. This is more from a general approach. But I think from a veterinary perspective, I'm very interested in the application of epigenetics into health. And are we able to get to that point where we can not only take a skin sample from a dolphin and determine the age, but actually also give us an indication of health status as well? So as part of this research, we also looked at some of the samples that my colleague, Dr. Lori Swacki, had been including in her VSOP model, so the Veterinary Expert Survival Outcome Probability, excuse me. And so basically this graph is looking at the likelihood of a dolphin being able to survive for 1 year, and that is on the y-axis. And then we were looking at the age residuals of the DNA on the x-axis. And so what's really interesting here is that the animals which showed a reduction in probability of survival were also the animals that showed an increase in age residual or potential age acceleration. So the reason that this is super interesting is because the VSOP score, or the probability of that animal surviving, was achieved through looking at physical exam findings, biochemistry, hematology, and determining profile effectively of how healthy or unhealthy that animal was, and subsequently how likely was it to survive. Whereas the DNA was not involved in that assessment at all, yet we were still able to reach the same conclusions of the animals that had a reduction in chance of survival. This was also depicted in their DNA. So I think we're still very much at the embryonic stages of being able to interpret epigenetics and health, but it was really exciting to be able to reach the same conclusion from the same samples from two very different perspectives. Similarly, we also looked at the effects of human interaction and entanglement in dolphins to see whether we could see that there was a increase in biological age in these animals as well. And so you can see that the fishing interaction is in red, and you can see that these animals were more likely to be the other side of that line. So remembering the guy with the walking stick, these were the animals that were potentially a little bit more compromised. So from a marine mammal stranding network perspective, what samples can we actually use for epigenetics? So we really focused our samples on live dolphin health assessments. We really wanted to get very high quality DNA to be able to establish this epigenetic clock in bottlenose dolphins. But I think it's also very possible to be using samples of DNA from deceased animals as well. So ideally code 2 or code 3 samples. I suspect once we get to code 4, we'd start to see some degradation in the DNA quality, and that might affect the accuracy of the results. So ultimately, epigenetics can be used to estimate age remotely, and I think it can give us a more general indication of health status. We recently published these findings, so you can scan the QR code here if you want to access that. And I would like to give a huge thank you to all of my co-authors and colleagues that have facilitated this research. It's really taken a village to get it to this point, and several years of hard work to be able to share this publication with you. For our future work, we're really interested in expanding the use of epigenetics into endangered species. So we recently applied this to the endangered Lahili's bottlenose dolphin in Brazil, and were able to predict the ages of animals that are in a longitudinal study photo ID program, but age information was not previously known for them. So this is really useful to the local biologists to be able to know, for example, which females are of reproductive capacity and which ones should be closely monitored for signs of a calf. We're also interested in applying epigenetics to unusual mortality events, stranding response, and seeing if we can explore the relationship further between epigenetics and health, including the exposure to PCBs. So as you can see, this has taken a lot of funding pots to make this happen. Side note, epigenetics is not cheap, unfortunately. That's one of the disadvantages of this research. But I would like to give a huge thank you to everybody that has been very supportive of this work and has provided us funding to be able to complete this. I'm happy to answer any questions, or please feel free to email me. Thank you.
Thank you, Ashley. That was great. We do have time for some questions, commissioners or scientific advisors. Yeah, Jason.
Thanks. That's really, really impressive results. I didn't realize how accurate it was. And I'm thinking about going about estimating an age structure of a population through epigenetics, and you have a really pretty tight age estimator, but you know, if you're building an age structure, you care more about sort of the precision in the early young animals. And I think my recollection is that your precision was better for younger animals and then becomes less precise as they get older, which is a good thing. Is that correct?
Yes, that's correct. I'm sure Laurie's disappointed that I put the 1.9 years on there as the average rather than breaking it down. It was just in the interest of time, but we were able to actually predict the age into just a median absolute error of just 0.3 years in animals that were less than 5. So in the really young animals, that's obviously what we're interested in, and especially when we're trying to dial in that when is the age of sexual maturity. So in the older animals, so over 25, we would see that the MAE was closer to 5 years, and that the 1.9 was the average across the full lifespan. So I think it's multifactorial as to why we see that decrease, and I won't take up your time by going into that, but it was definitely much more accurate in the younger age groups.
We have an online question?
Yeah,
maybe?
Andy's on screen above now.
Hey, Ashley, this is really lovely stuff. Thank you so much for the presentation. Very quick question. and you mentioned this briefly in your talk, but for species or clades in which we're never going to be able to develop a species-specific epigenetic clock, you mentioned the idea of an O'Donoghue clock. Can you just talk a little bit about that? I'm thinking about clades like beaked whales, for which we'll never have, animals of known age.
Well, you set me up for success with the beaked whale comment there, Andy, because we've actually already started doing epigenetics in beaked whales with Diane Claridge. We are collaborating with her. But you're right, we don't have sufficient samples to be able to necessarily create a beaked whale clock from known-aged individuals. I think the best that we can really hope to achieve in species like that is to have sufficient samples to try and validate a clock or to see which clock is likely to be the best fit. And so I think the really, really broad clocks of having an O'Donoghue clock, you're going to get a much wider error in the age estimate. So I think the best bet is probably to try and find a more closely related species, which is what we did with the Lahille's bottlenose dolphin, and then apply that clock. But the bonus is now that there's so many clocks that have been produced, there's no harm in applying all of the clocks and seeing which ones fit best if you have that small subset of samples that are of known age to try and validate your estimates.
Awesome. Thank you.
Thank you, Ashley, and thank you to all of the speakers this morning for a fascinating session. You'll have a chance to grill all of them. After our break, we want to start promptly at 11:15. Thank you all very much.
All right, just give us a moment as everybody gets in their seats, making sure if people are coming back online. Actually, let's give it 30 more seconds for the doors to close. And if we have our virtual panelists, if you turn on your video, we'll make sure we can see you in our room on the screen. Perfect, we can see you. And presumably, once you're off mute and ready, we'll be able to hear you as well. And with that, Randy, you can take it away.
Thank you, Brady. And I also want to thank the team of Lori and Lori Schwacke and Lori Leach, I knew that, for putting together the team of speakers this morning. You guys did a great job of bringing fantastic information together, so thank you. We've got 45 minutes for discussion. We can take the discussion in just about any direction we want to, but I think what Jason did yesterday was a very useful way to go in terms of first asking the speakers if they have any questions for one another. Cynthia, please.
Okay, great, thank you. Chris, I had a question, practical applications question, as we are all going to be downloading your 3D printing model and thinking about how each of our organizations can either partner with you or try to reproduce what you're doing. Very practical. Was your approach to take drone pilots and teach them marine mammal behavior, or was it to take marine mammal field biologists and teach them how to be drone pilots? And I know that's getting into the nitty-gritty of it, but just thinking about, like, how do we all now move forward, and what's the best approach to do that? Should we be finding drone pilots in our cities to work with us?
Yes, great question. I think finding drone pilots and teaching them sort of whale behavior is the quickest way to get to success. We've done a little bit of both, but typically it's harder to learn to be a good enough drone pilot than it is to kind of just spend the time required with whales and flying over whales. I like to kind of think of the kind of three main, most commonly used tools are kind of photogrammetry, blow collections, and then tagging now. And that's kind of the progression I would do. So if we're teaching someone new, we might say, do a lot of photogrammetry, it's high altitude, it's low risk, but you're seeing and following those behaviors, and then you can kind of work your way closer to the animal into tagging approaches. But one of the benefits also with the drone-based tagging is that kind of a failure doesn't cause a lot of impact to the animal. So if you kind of drop and miss, you can just pick up the tag and try again, and it's not the same kind of impact as to racing up on it with a boat. And so we might give kind of an unrealistic expectation of success. People are just like, okay, I'm going to get tons of tags on now, but we've kind of changed the game in terms of you might go out for a whole expedition and hope to get 2 tags on, and now we're getting 20 tags on. There's a middle range where you might not be the best pilot in the world, but you're still able to have success and get tags on and collect data. So long answer, but yeah, I think the best way is to find an already skilled pilot and start there.
I had a question for you as well. That impressive array of species that you've been able to put tags on so far. The gray whale skin is not the smoothest skin of any whale. And have you had problems with getting the suction cups to be able to attach and stay on as long as with the other whales?
I think actually the most difficult species has been fin whales. I think mostly because their speed that they travel at. But it's, again, so we're just kind of using off-the-shelf tags and we're trying not to have to modify anything more than we need to. So just using the suction cups that are provided to us. And we've done limited kind of side-by-side with drone tagging and pole tagging. And from what we've done so far, there's really no statistical difference between the duration of a pole tag and suction cup tag or drone-based tag. So yeah, different species are going to have different durations, of course, but it was nothing that was noticeably different from drone to pole. So.
Speakers in the back, speakers in the front. Yes, Ashley.
Ryan, I had a question for you. Excellent talk. I really enjoyed it. Predictably, my question is about methylation and aging, but I was just curious as to whether you had the proof of concept, if you had a known population of animals in a given area of known age demographic, whether you had been able to match that with the eDNA.
I'm so glad you asked, because the Slack chat that's happening in the background of my screen is our resident methylation expert who's like, oh, wow. So we didn't know about your paper, and now my whole group knows about your paper, and it's the same species. And we know how old one of those animals is. The Navy will not tell us the others, and they, in fact, won't tell us the names of the animals. Neither will they confirm the number of those animals, although we can see them with our eyeballs. So actually, so no, we haven't done it yet, but I now have many chats from the last hour that says, yes, here's how we're going to go do this, and we do know how many there are, despite the fact that that's unconfirmed officially, and they are different ages. So we think that we could, there's sort of a path to using your model in the field.
Awesome, thank you.
Aaron.
I guess follow up on that question, how fresh, so to speak, would the eDNA have to be in order to measure the methylation and thus use this technique?
Is that for me or for Ashley?
I have no clue.
I can tell you I have no clue either, but I do know that the methylation patterns are fairly durable. I think I'm looking to Ashley to confirm or disconfirm that. But the size of the fragments, you lose long fragments real fast, within hours. And so your best case scenario is very fresh sample that has nice long fragment size, and all those methyl groups will be on there. But I think, and Ashley, maybe ask you to confirm that even in more fragmented older DNA, that the methyl groups will have stayed on there for the most part.
Yeah, I agree. And I think from the DNA methylation with the aging perspective, I touched on it with the code of the animals. So I think the fresher the sample, the better the DNA. However, the storage time doesn't appear to be an issue. So we did include samples that the DNA had been extracted and then the DNA had been stored for 20 years, and we didn't have any issues with those predicted ages of those individuals. So I think as long as it's stored in good quality, then I think that you should be fine.
And the real killer app there, I think that one of the killer applications of this is maybe not obvious, right? Where there's the aging application where you're asking like, how old is this animal, like, or these particular animals, which is one great application. There's the application that I immediately leapt to, that's maybe less obvious, which is how many individuals are represented in this sample, where I've got a mix of ages. Can I sort out that mix of ages by trying to decompose that and say, oh, okay, well, I know there are at least 5 animals represented in this sample because they're 5 different ages, which that'd be a cool thing to do with that technology.
Daniel?
My question is for Ryan, and that what you just said dovetails into what I was going to ask. I appreciated the schematic you showed about organisms being discrete, and you showed a number of tuna or whales or whatever. And looking at the maps that you presented for the samples that you have for the West Coast for cetaceans, and then the maps that you showed for the fish at various depths, how do you account for, or how do you handle the swamping of something like fish that are much, much more abundant than cetaceans? When you're sampling, do you take a bottle of sample of water, and you'll have much more eDNA from organisms like plankton and fish than cetaceans, and how that is swamping, potentially swamping the cetacean signal there?
Yeah, great question. It's the magic of PCR primers is that they're doing 2 things at once. First they're selecting for species of interest depending on your primer design. So in that case, we're using PCR primers that are specific to vertebrates. So what that's doing is leaving behind the vast majority of DNA in the water that's bacterial and viral, archaeal, and so forth. And we're saying, hey, we actually don't want to see the vast majority of DNA in the world. We are interested in the needle in the haystack. Just show us the vertebrates. And then so the PCR primers they're playing 2 roles. They're doing that selection, and then they're helping amplify it. So then we blow up that needle in the haystack. Now it's a mixed metaphor. I'm not sure where the metaphor is going, but that we make a much bigger needle. Like we have a a much amplified signal that we can sequence. We can map species to. We can look at concentrations of species-specific DNA. And it actually doesn't matter how big the haystack was. It doesn't matter how many bacteria were in there because we can zoom in on the things we care about. And in our case, the Navy is particularly interested in beaked whales because they're hard to otherwise study, as was mentioned a couple of times, I think, earlier. And so I counted over the break, we've got 27 beaked whale detections from these samples and so that's kind of the ultimate needle in the haystack. Like these are samples that were taken, 2 liters of water, taken for a totally independent purpose, never seeing the whales at a grid of locations at a systematic, not a opportunistic sample, and we can still see beaked whales even. So I think that answers the question.
And I'll also note for those in the room that Mike W. asked a similar question that Ryan dovetailed the answer, I think, perfectly already. So I'm not going to repeat the question. Ryan, if you want to look in the answered questions later, we can figure out if that was correct in my assumption.
Yeah, just to follow up on the last conversation about the methylation. Do you think, as you're working on these models to eventually get to abundance from the eDNA, do you think it's going to be necessary to understand the ages of the animals? And the reason I ask is a conversation I was having with Jason at the break about, is there potentially different shed rates of skin and eDNA of different age classes, and is that going to be important? And maybe this is something Cynthia can chime in on too, as she's probably dealt with a lot of different shedding animals.
Sure. There are a couple of parts of that. To the age question, does age structure matter in stock assessments? I think I'm probably talking to a room full of people who know more about stock assessments than I do. So I'm going to go with yes. Probably you want that age structure if you can get it, because it's going to be useful for demographic models and for stock assessments. To the specific question about abundance, how do you map abundance of the DNA, the concentration of a species in the world, onto the abundance of like a number of animals? And that, as I sort of mentioned, that what you got to do is you need some external source of data to calibrate against, right? So it could be visual, it could be acoustic, it could be whatever, historical records, but something to get you out of just looking at molecules and calibrate against the unit of, we actually think of it as sort of a units question, right? Like, are you interested in kilograms? Are you interested in N individuals? But to convert units across, and every species, like you said, maybe have a different shedding rate, or maybe Tuesday sheds more DNA than on a Wednesday. That's why we don't do this with one sample. We do this over and over again across space and time. With that replication, we have a sense of that variability, and we know what the mean is, as opposed to the variance. It gives us a sense of signal versus noise, and to be able to calibrate that to your units of interest, which may be N individuals, it may be kilograms if you're a fisheries person, and so forth. But it's not one answer. We're never going to be able to say, okay, here's 100 molecules of DNA that equals one whale. Like that is not true, and it's not going to be true. It's that there's a dynamic relationship, and you're looking at an average over time, and you want to be able to do that in a robust statistical way.
Yeah, and Ryan, I think I would add not to the age question, but other things that could impact the shed rate that we should be thinking about, like health and environmental conditions. So what about the salinity of the water and the changes of salinity and where the animal's moving? And will that be impacting the shed rate? And then health status, we know certain cetaceans actively exfoliate, and when they get sick, they might not be exfoliating as often. So will that be impacting it? That's getting into probably a finer scale of change when you're thinking about the shedding, but it is something to consider. And how could we be maybe including that in the way that we're modeling and analyzing the data?
Aaron.
Yeah, thank you. A question for Cynthia. So you're talking clearly about using your blood sampling technique remotely to collect things from animals in the field. So there'll probably be a certain interval of time between when the sample's collected and then when you can retrieve it. I was curious, I'm not an expert in this by any means, but do you need some sort of preservative, or is there a time scale over which some of these parameters would be expected to change in the blood sample before you'd be able to collect it on the time scale of minutes, hours, days? I don't know.
Yes. So so the preservative that you put inside the tube, that collection tube, is important when you're thinking about what do I actually want to measure. And one of the things that we want to be hearing from the community is, what do you want to measure, so that we can be doing the studies to look to see how hard is it going to be to measure cortisol? How hard is it going to be to measure progesterone? And some of these things are much more stable over time, and some aren't. So with cortisol, we're going to have to really be thinking about deployment of a tag and preservation of that sample, and then getting it back quickly. So it's got to be waterproofed, it's got to float, it's got to have some kind of a beacon of sorts, so that we can quickly retrieve it. Or maybe if we're measuring cortisol, we're going to have to do it from a tag. And maybe we partner with Randy on like a tadpole type of solution. So absolutely, we're going to have to be thinking about time, but there are ways to mitigate that and to account for that. And so it really comes down to, we care about things that help us understand health, like hematocrit or PCV and white blood cell count, but we also want to now hear from what are the questions that you have for the different species so that we can start looking to see how exactly are we going to design this to get at those critical parameters and questions.
D.
Yeah, my question is for Ashley. I'm curious, well, there's kind of one quick question. Are you, for the DNA, are you only using skin or are you using other tissues for epigenetics? And then to Andy's point, particularly with the beaked whales, do you see an opportunity with using any of the biological collections, particularly museum collections, where you have had tooth ages for individuals, and you probably have a suite of samples that are taken, skins usually taken too, and either archived, frozen, or in other in other ways. But is that something that would be beneficial, even if it's not a current living animals? If they're older animals, does that feed into understanding the biological clock for a species that you'd be able to apply to current living animals?
Thanks, Dee. Great questions. To answer your first one, for bottlenose dolphins, we started with blood, and those were the known age Navy dolphin samples, and that was really for proof of concept. I think it's fair to say that the quality of the DNA from a blood sample is better than from a skin sample because of the conversations that we were just having with the sloughing and the changes in the skin. We did decide to move forward with skin primarily because of the remote biopsy perspective and the application of that to large non-tractable cetaceans. I think for the skin samples, we have found that there's more accuracy with a full thickness epidermal biopsy, such as what would be taken if you were wanting a blubber sample. Getting that full thickness of the skin is more accurate than just a skin scraping, because then you're potentially just getting the decaying epithelial cells from the surface. So I think depending on how detailed and into the nitty-gritty you want to go, depends on kind of the level of the sample. In different species, I think it varies as to what tissue you want to do. So we're currently in the process of writing up the first epigenetic clock for northern elephant seals, and we actually did blood samples for these animals because they haul out and they were having tags attached to them anyway, and blood samples were being taken. It made more sense to use blood than it would to use skin. And so I think it can be species specific. I think the key is to have a large number of samples for that specific tissue that you decide to move forward with. And then to answer your second question, I think absolutely using archived tissues and, in species where we have very little information, having some sort of a life stage estimate, be that from a tooth estimate or from baleen or from racemization, something that is giving you kind of a ballpark figure, then that could help to dial in the clock and calibrate the clock. One of the challenges, I think, with the biological aging is that ideally you want the known chronological age to be able to then look at the age residual, i.e. the difference between the DNA predicted age and the chronological age, to give you that inference for age acceleration or biological age. So if you don't have an accurate chronological age, then you are basing an estimate off of an estimate, if that makes sense. So I think that would reduce your accuracy. But again, depending on the status of the species, having some information is definitely better than no information at all. So you've kind of got your gold standard of what we can do, which is honestly why we went with bottlenose dolphins as our pilot species to do it with, because we had the most availability for known aged samples. But I think there is absolutely application for other species with varying amounts of knowledge, and diving into the museum archives would be a goldmine for sure. It would be really interesting.
Peter.
Yeah, I have one of those questions which I don't really know how to ask, but focusing on the stock assessment theme of these several days, I've heard, from all your talks, Michelle, Ryan, Ashley, Cynthia, Chris, things about detecting the presence, are marine mammals there or not, some on age, thinking about getting the age distributions, looking at individual or in some sort of extrapolating to population health, seasonal success. Michelle, you did that with the gray seals, and sort of breeding success. So I'm just sort of thinking in that matrix of what we need for stock assessments. And it can't just be for you guys, it could be for the audience. You know, what are you seeing that's coming out that we ought to, the directions we ought to be going to help us with stock assessments? I think each of you have mentioned different things in different aspects of your work. And I don't really have that way to fill in that matrix, other than it was really cool to see the map of distribution on the West Coast and then compare that to fish, what we can get for fish and what we can get. It's really cool to see the seasonal differences up in Canada of the gray seals. It's really cool to talk about epigenetics giving us age classes and going from general to individuals and back to general. So that's not a question, it's just kind of where do we go from here, and what should the commission be thinking about doing? Are there particular avenues of work, and you can say it now or you can think about it, avenues of work that'll get us from this health-related and genetics-related thing into pushing better methods for stock assessment? I know that's the challenge y'all got, and you've done the best to answer it, so what can I say?
Anyone want to expand on that? Cynthia, sure.
I think it's a super important question, Peter, and yes, we are all thinking about it. You know, I think of all the things we're hearing, one thing that is always nice is if human medicine is ahead of us and if we're chasing them. When we're ahead of them, it's harder and it goes slower. So, you know, when I think about epigenetics in particular, and this whole biological aging thing, I'm just so excited if we can actually start to make progress in that area, but it's going to take a lot of an investment of resources to do that. In samples and the technology, we need to, hopefully it'll get less expensive, but trying to, you know, being able to take a sample from a free swimming animal and then make conclusions about its biological age, like how the pressures of its environment are actually impacting that individual animal, and then piece that together with population. I mean, I just think there's so much potential there, but we have a lot of work. We're far away from being able to make strong conclusions, but the human medical field is doing it. So if we can focus on that, I think that's I wouldn't call it a low-hanging fruit, but I would call it one that has a ton of potential for answering the kinds of questions we want to be answering. That's just one thing, Peter, that comes to my mind right away.
So Ashley, a question for you. Do you see anything on the horizon that might bring down the costs of doing epigenetics and make it more feasible for researchers?
Short answer would be no. Would I ideally like that? Absolutely. I think that the only thing that might facilitate that is that, as you saw in the presentation, it is garnering more and more interest and support. And I mean, even just from, you know, Laurie and my first conversations about trying to do epigenetics 5 or 6 years ago, you know, we applied for several grants that we were not successful in because we got the response of, well, you don't know that this is possible. And even though it was possible from a human perspective back in 2010, for us to be applying for grants for in 2018 and 2019 for marine mammals, we were met with closed doors. And I think that, you know, 5, 6 years down the line, we're definitely not at that point where we're being challenged of, is it possible? So I think the momentum is there. And so ideally, I would like to see that more funding is headed in that direction. The only way I think at the moment that we could really bring the costs down is if we did a more targeted approach. So right now we are doing the full 37,000 CPG sites on the mammalian array. You could pick out, you know, 3 or 4 specific genes and associated CPG sites and hone in on that. My personal caution with that is that there's so much that we don't know yet, as Cynthia just alluded to with marine mammals. And in the human field, we're really seeing these advances where we can see, you know, specific methylation sites for Alzheimer's disease or for diabetes. And, you know, we're not even getting there at all with marine mammals at this point. So by continually running the 37,000 sites on our samples, I'm excited for where we're going to be in 5 years' time, that we have that data archive and we can go back to those samples and hopefully learn even more about them. So will the cost of the individual sample go down from the Clock Foundation's perspective? I dare say probably not. It's currently $160 per sample, which is not huge when you're comparing it to like PCB analyses or something, but it does add up when you're submitting plates of 96 samples each time. Yeah.
Jason.
A question for Michelle. I think it's been, what, 4 or 5 years since you published your paper about measuring gray seals with the UAS system. And I remember just being shocked that, you know, when you think about what's involved in capturing seals and weighing them on a rookery, that this would be revolutionary. And maybe I'd like to know if other groups with other species have adopted the method and applied it, and, you know, if so, who, and if not, what do you think might be barriers? Is it, I'm sure there's interest, or maybe it's just investment in the techniques and something like that.
Yeah, I talked with some graduate students who were interested in trying to apply it to like Hawaiian monk seals. I don't know exactly how that went. And I've been talking with a bunch of people up at NOAA who are interested in using it for ice seals. So I think you'd have to definitely adapt the methods for some of these different species. And there's definitely the time investment and the additional permitting for all the drone work. But I do think it's definitely very possible. And I've been really astounded just how tweaking little things about the methods, you know, just tweaking your camera angles and some of the processing workflows, how big a difference that makes. So I think it's definitely something that I'd love to see adapted to other species. And I'm definitely very happy to help anyone who wants to try.
Francis?
Question for Ryan. You've talked a lot about eDNA, but you had a little tantalizing slide or some words on a slide about RNA, but we didn't talk about it, or you didn't sort of go into it much. So I'm curious about where we are with using RNA, and then, because that to me would get us down a pathway of picking up changes like, I mean, reproduction or inflammation or some of the physiological processes that were other than just presence or absence of the animal. So could you maybe enlighten us a bit on where we are with using RNA?
Sure.
On water samples?
Yeah, we've started into that example, into that adventure a little bit. And the example I had in my slide was the decay curve for two different kinds of RNA. There was a messenger RNA and a ribosomal RNA. The messenger RNA goes away, sort of as you might expect, goes away immediately. And so it was like within, it might have been an hour or a couple of hours, we couldn't see the signal anymore. And those were from water samples where we were almost right on, you know, close to the dolphins, right? We were very close. So in the field, it felt like our conclusion was we weren't going to be able to go out, grab a random water sample from the world and expect to see dolphin mRNA because it just won't be there. The ribosomal RNA will be. So, but, you know, if you're in a situation where you do have, you know, for example, captive animals or animals where you have eyes on them and you're close enough to them, you can get RNA out of the water, and you can get you know, functional genomics out of that if you're interested in you know, for example, you mentioned you know, reproductive status or whatever, any gene of interest, the expression of, it will be there. It'll be there at a very low level, and with DNA with DNA or RNA in the environment, you know, there's this enormous dilution factor of the ocean. So, but it will be there. And so where we've been able to get a hold of it, we've actually done that in salmon. Aidan Ip is the staff scientist working on this. You can do it with long-range sequencing using a MinION, and that way you're not amplifying. You're sort of shotgun sequencing everything that's there, and you can take a look at the expression of particular genes of interest. So that's nice. You're not touching the animals. You know, it's non-invasive, but it is probably limited to that situation, I think. I guess while I have the mic on, in terms of lowering costs of methylation questions and analyses. I think we've had some reasonable luck with those MinION devices as well, and those tend to be low cost. You don't have to do the bisulfate sequencing. That's a big pain. And I know you guys actually have a fancy chip, so maybe it's a different thing. But I think there's some hope for reducing costs. And then finally, as to the stock assessment question that Peter started out with, we have just now in March the NOAA's Hake the Hake stock assessment for the first time has now integrated eDNA data into their stock assessment as part of their ensemble model. And so going forward, it'll join into those models in the future. And so that's my answer to that question of like what are the steps? That's the step is you start adding this as part of a larger multi data stream model. And then you see how it behaves, right? And after a while, you develop confidence in it, or you say, okay, it's good for this and not good for this, or here's how it's adding value, and go from there. But it's not a one-time thing.
Robert?
Yeah, thanks. First, everybody, I'd like to say thanks for your presentations. They're really, really cool. What people are doing and creating new approaches to things. Peter's question about how these different new approaches might be helpful for stock assessments and the data that are in stock assessments, that one of the things that comes to mind is a question for Ryan, that in some cases stocks are being, the population size of stocks is being estimated using mark recapture, genetic mark recapture. And so my impression from eDNA to date is that the DNA degrades so quickly that you can't get enough DNA to actually identify individuals. Ryan, one of your slides mentioned haplotypes, and obviously that's not individuals, but it kind of tells you something about the stock. What do you know about being able to identify individual animals from eDNA with the idea of possibly using a mark recapture approach to estimate population size?
Boy, that is, I think that's a deep hole that I don't want to dominate the conversation here. I'll just say I don't know. We haven't done it. But I think the hurdles are, as you say, right, you got to be pretty close to the animals in order to get enough genetic material that it'd be useful. And if you're that close to the animals, you probably don't need the DNA, for that reason. You might be able to count them. But we do have a project looking at, Pedro Brandao is leading this work, funded by the ONR, looking at haplotypes within again, the dolphins. And we do recover individual haplotypes. You are able to track those. And so it is useful in that respect for looking at number of contributors to a sample. But again, I think there's this dilution problem where, once you're too far away from the animals, it's just going to be such a rare signal. I think the noise is going to dominate the signal.
Karen?
Yeah, thanks. I had a different question for Ashley, but I just wanted to follow up on that. I mean, one species where there could be potential, which is notoriously difficult to assess in some areas, like on the West Coast, is the harbor porpoise. You can be surrounded by harbor porpoise and not be able to get any DNA for any functional work because they're so challenging to sample. But if you could get reliable repeat sampling, you may be able to do some sort of a mark recapture for some of the localized populations that we have. And I'm totally thinking off the cuff there, but it's an intriguing possibility. My main question was for Ashley. I was curious what the time horizon is for the divergence between chronological age and biological age. In other words, how soon would you start seeing that change? Does it take years, or is it something that perhaps could help inform? One aspect of stock assessments is assessing mortality and serious injury, and obviously things that have sublethal impacts on animals do have a long-term serious injury component that is very challenging to assess. And I'm wondering whether there's potential, if there are entangled whales that we can't disentangle for whatever reason, or that we see over time at various points in time, would you be able to detect a divergence, or does it take a multi-year time horizon for that?
That's a great question. My understanding is that the chronicity would impact how well you're able to see that divergence and actually see the age acceleration taking place. So for an example, like in a human, if you broke a leg, that's not necessarily going to impact your DNA methylation profile. But if you've got chronic liver disease, then it's going to be something that is then reflected in that. So I think from a large whale disentanglement, unfortunately, we do see that these cases are often very chronic. And so I think the slide I shared in the presentation where we saw the entanglement cases in the dolphins, we didn't kind of dive into the duration of entanglements in those specific cases. I don't know that it was necessarily always known, but we were starting to see the age acceleration, and some of those animals were really quite young. And I do remember a specific case that Randy had in Sarasota where it was a young dolphin that had been entangled and had been separated from its mom at an earlier age, and Randy was able to handle it at multiple health assessments, so we were able to sample it over time, and we did actually see changes in its pectoral flipper radiographs that we wouldn't necessarily have expected for such a young age animal, that it was actually showing that it wasn't developing at the rate that we would expect for that individual. And the entanglement there was likely the stressor or something that could have impacted it. So I think the more chronic the injury, the more likely we would see it reflected in the methylation profile.
Ashley, is there utility to using multiple age determination techniques when you have the opportunity, like the PEC radiographs, in addition to the methylation, or does it just get messy?
I think I'm biased to answer that, but I think if we don't have the life history information, so if it is an individual where you don't know when it was actually born, then I don't see there being any harm in performing multiple methods and trying to give ourselves the best chance possible at obtaining the most accurate age estimate. I think for the younger individuals, as we talked about earlier, the epigenetic aging is a lot more accurate in the younger individuals, so I would feel a lot more confident. But I think if you were looking at an animal that was over 25, then having more methods might be of benefit.
Lori?
Okay, so I'm going to get us off on a kind of crazy tangent. This is a question for Randy and Chris. So you both had great presentations on super significant advances in the tagging. Do you ever see it being the case, or you see combining your efforts such that, using some of the smaller drones and going after some of the smaller cetaceans, but trying to get these pin tags rather than just suction cup tags, and looking at what the military can do with drones, with onboard guidance systems and whatnot? Do you ever see that there would be a drone that's kind of a combination of pilot and automated guidance?
There is an SBIR currently for limpet tags and type C, like the implantable tags. I don't know how much I can say about it right now, but that's something that is being worked on and funded by ONR.
And it would be great if that could happen, because everything that has to come together now in terms of getting the animal to ride at the bow of the boat and that sort of thing, you wouldn't have to worry about if you could just go out and take any surfacing dolphin. I think there still would be issues with dolphins detecting drones and avoiding them. And just given the minutia of the timing of getting a tag on a fin now with an experienced tagger trying to do it from a drone would be really challenging unless you had the guidance support that is way beyond anything that I know anything about. But you'd get so many more tags out.
Yeah, and if I may just add, we did have a project funded by the U.S. Trade Representative Office where we were leveraging military technology to incorporate into a drone for specifically looking for the smallest of cetaceans. And so we were making progress on that, but that funding has been cut. So if it gets turned back on at some point in the future, we'll pick back up where we left off. But there, thankfully, is a way to declassify some of those assets and resources and incorporate them for conservation. And so hopefully that will get turned back on and we'll be able to report out.
Aaron?
Thank you. I'd like to ask a question to Michelle. I really enjoyed your work and presentation on measuring respiration rate and temperature with cameras. Maybe I have an odd fixation with shining lasers on marine mammals, but I was wondering if you had any comments on the practicality and feasibility using a laser Doppler vibrometer to make some of these measurements. They can sample much higher rates in time. And I mean, you, obviously, you don't want to point to the face, obviously, but if you probably could point at other parts of the anatomy and maybe get a cleaner signal.
I don't know specifically about using that instrumentation. I think there's some potential with like LiDAR, and that there's been some recent work in some terrestrial species where they've gotten respiration rates using LiDAR. I don't think you get heart rate from that. But yeah, so I think there's definitely some other applications. If I don't know specifically for lasers like you're talking about, but
Okay, thank you.
Aaron?
A couple other ideas I've been getting excited about with applying some of this tech to SARS would be combining methylation for aging and hormones that's either collected with a blood sample or with a snot bot. And if you do that in lesser studied populations, you could get perhaps at a better sense of reproduction rate for specifically viable females. And another idea I had was using methylation techniques on prey samples collected from gut contents or fecal analysis to get an age structure of prey. So if you guys want to speak about the potential of either of those applications for SARS, that'd be cool.
Yeah, very cool ideas. I really like the prey one. We've certainly talked regarding the hormone and the reproductive question. It's something that's very important to me. I would really like to be able to get to that answer through the remote biopsy sample. And we are fortunate in the database that we have. Most of these animals had a full physical health assessment. So we had ultrasound information which confirmed if the animal was pregnant or not. We also have blood progesterone, and so now we have the epigenetics from those. So it's definitely a question that we are keen to explore further and to try and answer. We don't have the answer as yet. One of the challenges is the timing of the health assessments with the reproductive timing. We often either get the very early pregnancies, or we have the sort of third trimester, which we don't want to do a lot with, and they're going to get let go pretty quickly. So we've been working diligently to increase the sample size across the full gestation period to try and see if we can get at that. So back to the chronicity question with the DNA methylation, I think the very early pregnant females, there may not have been enough time for us to be able to detect that in their epigenetics. So we need more samples throughout the duration of gestation. But to be able to combine that with the hematag results would be fantastic.
We've been given a couple of extra minutes beyond the 12 o'clock agenda cutoff. If we have more questions. Peter?
Just to sort of follow up on another area that when we review stock assessments, we note that many stocks have not been assessed, particularly out in the far Pacific Islands, remote areas. So again, Ryan, your detections of beaked whales were really interesting, but I just throw that in as another area to think about as you ask what these technologies can do to help us in stock assessment. There are a lot, I forget the percentage that haven't been assessed, but it's 30% of U.S. marine mammal stocks have not been assessed due to remoteness, difficulty to see, rarity, whatever it may be. So that's just another parameter to throw in there to try to think about. Obviously, eDNA gave us a little really interesting picture on that. And yesterday we talked about satellites, but just put that one in the hopper.
Brady.
Yeah, just to report on a couple things in the virtual Q&A. Ryan, thank you for answering some of those questions. So in a second, after I ask two more, maybe if you want to comment on any of the things that you found, of note that you answered online for those of us in the room. Two quick questions for Chris. One is how long do some of your tags or suction cup tags adhere for? And then the other question is, would it be possible to collect a genetic sample from a suction cup tag? So I think those are pretty quick. And then there's one more for Ashley, maybe, but I'll throw it to Ryan since maybe he's been thinking about it. So Chris, you first.
Yeah, so for how long the tags stay on, typical suction cup tags, your maximum is sort of around 24 hours. Logistically, you might not want them on that long if a whale swims far away, 100 miles in a day or something, and you have to go pick up the tag. So again, with the limited pole tagging and drone tagging we've done side by side, the durations are pretty similar. So the timing is more dependent on logistics than maximums. On the genetics, we have been doing a lot of swabbing the suction cups when we retrieve them for using that DNA to sex the animals, which has been really successful for, I think we verified it with right whales and have been using it with humpback whales. But I don't know the extent to which the quality of that DNA can be used for other things. A lot of times we do get bits of skin that will get sucked up into the suction cups. So that would be a little more high quality than just a cotton swab on the suction cups, but it'd be interesting to dive into what can be extrapolated from those samples.
Okay, so then Ryan, do you want to comment on anything you've answered online? And if not, I'll ask a question for Ashley.
No, I think I'll pass. You've heard plenty from me.
All right, so Ashley, this question is interested if you can say more about if or how the age residual may reflect the health of individuals. Is it somewhat of a coarse scale, such as poor or good health, or is there a finer resolution possible?
I think at this stage we're still in the more broad category, whether we're just seeing a positive age residual or not. So determining if there is a degree of age acceleration rather than dialing it into the more specifics. I think the graph that I presented, which demonstrated the comparison with the high age residuals and the low VSOP score, which was the probability of the animal's survival, I think that's giving a more dialed in example of the health of the individual. So the fact that we could correlate with that, I think, is really promising, but it's still more a zoomed out, generalized health level at this stage.
And with that, I think we're going to bring an end to this session. I'd like to thank all of the speakers very much for your initial presentations and for the response to the questions and for all the great work that you guys do. And thank you for the great questions from the audience. We'll be back here at 1:30.
Okay, good afternoon, everyone. We've had two excellent sessions.